Welcome everybody to chapter 24. We're going to start talking about the urinary system, and let's talk about how this came about. Okay, so when animals were first evolving to, come from water to land, the process really became difficult by way of dehydration. Water animals, being aquatic bound, are not really at risk of dehydrating. But as they begin to spend more and more time on land, they needed, physiological mechanisms to conserve water. And the organ system that really does this the most for us is the urinary system. And we're going to talk about these organs as far as how they excrete. That means that they remove waste from the body. Specifically, how the urinary system is going to clean the blood, basically, of metabolic wastes. These are going to be produced by, the cells of the body that basically can't be used for anything else. So as far as our focus goes on the urinary system, you're going to see the paired kidneys and the urinary tract. Okay, and the kidneys are going to work to filter the blood to remove those metabolic wastes, and then they're going to modify the resulting fluid, which really allows these organs to maintain all sorts of homeostatic balances: fluid balance, electrolyte balance, acid base balance, and even blood pressure homeostasis. The the urinary system is going to produce a fluid that consists of water, electrolytes, and metabolic wastes called urine. And this urine is going to be transported, stored, and eventually eliminated from the body through these organ systems called the urinary tract. Um, First, we're going to look at the basic structures, and then we're going to look at the functional roles of the kidneys themselves. So I just talked about the kidneys. The urinary tract itself is going to be composed of those pairs of ureters, one from each kidney, that lead to a single urinary bladder and then another single urethra. Uh, this is either going to transport, store, or eliminate that waste, that urine, um, that basically just transports it from the kidney to the environment. And here you can see a figure that shows the organs of the urinary system in a female human body. And you can see we've sort of reflected the peritoneum and removed those intestinal organs so that you can get a clear view of these kidneys, which are typically attached to that rear body wall or very close to it, and hanging out very low in the lumbar region. And you can see each kidney with a adrenal cortex sitting on top of that adrenal gland. And then down here, you see a ureter leading to a bladder, and then this little spout coming off of the bladder is the urethra. And for females, that's going to be very short, and for males, it is the length of the penis. Now, the urinary system is going to regulate homeostasis, so let's look at what the kidneys do and how they do it. So the kidneys are going to remove metabolic wastes by filtering the blood, and then they're going to eliminate those wastes via urine. Okay, they're going to maintain fluid and electrolyte balance by maintaining that blood solute concentration, otherwise known as osmolarity, basically how much solute is in the fluid of the blood. And they're going to do this by either conserving or eliminating water or electrolytes like sodium, potassium, and calcium. They're also going to assist in the long-term maintenance of blood pH by either keeping hydrogen ions or eliminating bicarbonate ions. So they're either keeping acids or eliminating them, or keeping bases or eliminating them. And all of this is as that stuff has been used through the blood already. It also helps to directly influence blood pressure. Remember, as the blood volume increases, blood pressure also increases. So if the kidneys are able to pull extra water out of the blood, they're going to lower blood pressure. They're also going to secrete an enzyme that influences both blood volume and peripheral resistance. The kidneys also regulate red blood cell production in the bone marrow by producing that hormone erythropoietin that we talked about earlier. And they do perform a varying amount of metabolic roles like detoxifying the blood, activating vitamin D, and they also do that gluconeogenesis, producing new glucose that the body needs. And both kidneys are basically going to be the same shape. They're going to be the same shape and the color as basically those kidney beans that you've seen. That's where they get their name. That's where the beans get their name. Both kidneys are going to be outside of and behind the the peritoneal membrane. And so that makes these organs retroperitoneal. And typically, because of how the liver is positioned in that area, the right kidney tends to be a little bit lower in the body. And each kidney, of course, has that adrenal gland sitting on top, a major component of the endocrine system. And they're typically located between T12 and L3. And again, here you can just see that image. So again, you have kidney, adrenal gland, ureter, bladder, urethra, and each of these has its own sort of function. Creation of urine, transport of urine, storage of urine, and then transport out of the body into the environment. And again, you can just see on this posterior view here, you can see the left kidney sits a little higher than the right kidney, and that's because typically there's a big old liver here. Now, the kidneys are going to be held in place on that rear body wall, and they're going to be protected by 3 layers of external connective tissue. From superficial to deep, they are the renal fascia, and the fascia layer is really just dense, irregular, collagenous connective tissue, and it's going to anchor each kidney to the peritoneum and to the fascia covering those muscles on the rear abdominal wall. You also have the adipose capsule, and this is going to be the middle and most thick layer. Uh, this consists of adipose tissue that sort of wedges the kidney in place and basically shields it from physical shock. If a person undergoes some prolonged starvation, the body will typically use the adipose capsule's fatty acids as a fuel, and this causes the kidney to drop. And this is a condition called nephroptosis. Basically, just means drooping of the kidney. And then you have the renal capsule. This is the most deep layer, but it is also extremely thin, dense, irregular collagenous tissue that covers each exterior of each kidney, sort of like saran wrap. And it tries to prevent the kidney from becoming infected, and it protects it from physical trauma. And here you can see some of those layers. Remember, we're looking at a transverse section, inferior view here. So we're looking toward the head. And at the very, very covering of the kidney here, you can see that renal capsule. And this fatty tissue here, you can see the adipose capsule. And then on the outside, you see the renal fascia, which, again, if you follow that down, it connects it to this posterior abdominal wall, as well as the peritoneum in the front, the anterior portion. Now, a naked kidney basically weighs the same as a bar of soap, about 150 grams. And on the medial surface, what you're going to find is a sort of opening. It's this sort of C shape on the medial side of the kidney. And this is going to be where the renal artery, the renal vein, renal nerves, and the ureter all enter and exit the kidney. This hilum or hilum is going to open to a central cavity called the renal sinus. And the renal sinus is lined by the renal capsule, and it is filled with urine draining structures as well as adipose tissue. This connective tissue is basically going to serve to anchor the blood vessels, the ureter, and the nerves all in place. Now, this says these are viewable microscopically, but honestly, you can see them with the naked eye. There are three distinct regions. There is the outermost renal cortex. You have the renal medulla in the middle, and then on the inside, you have the renal pelvis. And each of these together, the renal cortex and the renal medulla are going to make up most of the urine forming portion, and the renal pelvis, along with several associated structures, are going to drain created urine that the cortex and medulla have then formed. And the renal cortex is really going to be basically reddish brown, and that's because it has such a rich blood supply. It houses about 90 to 95% of the blood vessels that enter the kidney. And at specific points, there are extensions of the renal cortex called renal columns that pass through the medulla toward the renal pelvis. And these columns house more blood vessels that branch off from the renal artery, basically as they travel to the outer portion of the cortex. You're also going to find renal pyramids, and these renal pyramids are separated from one another by a renal column on each side. I want you guys, when we look at this photo up here, to notice that the renal pyramids are going to be much darker in color, and they actually appear striped, showing that they are made up of parallel bundles of small tubes with fewer blood vessels than the renal cortex. Now, the renal cortex and the medulla of each kidney are going to contain over 1 million filtering structures called nephrons. These nephrons are the basic functional unit of the kidney, and each one is capable of filtering blood and producing urine. We're going to look at the basic structure of a nephron here in a minute, but it basically makes up two components. You have a globe-shaped renal corpuscle, and then you have a long snaking tube called the renal tubule. Now, some of these components are going to be arranged in the kidney in the renal cortex, with varying amounts of the renal tubule dipping down into the renal medulla. And the tip of each renal pyramid is going to taper into a slender papilla. You guys have remembered the term papilla. This papilla is going to border the first urine draining structure, a cup-shaped tube called the minor calyx. And urine is going to form, urine from 3 to 4 minor calyces are going to drain into a larger calyx called the major calyx. And then once you have 2 or 3 major calyces draining, they're going to drain into the large collecting chamber that is called the renal pelvis. And the renal pelvis is going to drain into the ureter is going to drain into the bladder. Now, there are going to be smooth muscle tissues in the walls of each of these calyces and the renal pelvis that contract and help propel urine toward the ureter. Now, and both of the calyces and the renal pelvis live inside the renal sinus. So let's look at this photo here. You should see, first off, the renal cortex, this exterior layer, renal cortex. Then you have these renal pyramids here. That's a really bad pyramid, but I'm taking some artistic liberty here. And this is going to be the renal medulla. You also have these renal columns here. And you'll see that each renal column separates each renal pyramid. You see one here, and here, and here. I'm not going to continue on, but you get it. And then you have minor calyces like this one here, and a major calyx, another minor calyx here, and 2 of those are going to drain into what is a major calyx, and then major calyces drain into this big open area called the renal pelvis. And the renal pelvis is basically just this big empty lumen of the renal sinus. Or I'm sorry, this would be the renal sinus, and it's filled with that fatty adipose tissue. But let's look at the nephron here. If we look over here on the right side of the page, you're going to see that it's made up of this globular renal corpuscle and then this long extension of tube, some of which you see dips down into the pyramid, into the medulla of the kidney. You actually see two different types of nephrons here. You see this one on the left. You see it just dips a little bit into the medulla, and then if you look at this one on the right, you see it has a corpuscle, but it has a much longer tube dipping into the pyramid, right? So that actually shows the two different types of nephrons, and we'll get into that in just a second. But I just want you to keep that in mind as we keep moving forward. Also, on the far left side of the image here, you can see this renal hilum. It's basically just the input area of the kidney. It receives all the blood vessels and the ureter and the any nerves that innervate this area. Now, every minute, about 1/4 of the blood that comes from the heart, about 1,200 liters per minute, come through the right and left renal arteries. Both of these branch off of the abdominal aorta. These renal arteries are then going to fan out into renal arterioles, basically, uh, that pass through the renal sinus and into the renal columns and the cortex. We're going to look at this order from largest to smallest as they make their way through. First, you have the renal artery, then you have a segmental artery, then the interlobal artery, arcuate artery, and the interlobular artery, otherwise known as the cortical radiate artery. So let's look at that image. All right, so first, let's get a good color that'll show up. Let's say yellow. Let's say green. So first you see you have a renal artery here, and then you see it branches and begins to get smaller. So you have the segmental, and you get even smaller up here. You have the interlobar, and then once you get even smaller into sort of this capillary structure almost, you have the arcuate artery. The arcuate artery is going to branch off into the interlobar artery. The interlobar artery is going to be branching into this afferent arteriole in the capillary here. It's going to go through the glomerulus, and it's going to come out the efferent arteriole here into some more peritubular capillaries, and eventually become an interlobar vein, arcuate vein, and it's just going to go backwards. So again, you're going to see renal artery, renal vein. They're the biggest structures, but basically they're just, it goes 1, 2, 3, 4, 5, 5, 4, 3, 2, 1. That's the basic structure there. Speaking of the blood vessels, you guys remember that capillary beds are going to be fed by arterioles and drained by venules. And in the kidneys, we're going to find an unusual capillary bed that you just saw that is both fed and drained by arterioles. So that efferent arteriole and the afferent arteriole. And the afferent arteriole feeds that ball-shaped capillary bed called the glomerulus. And the glomerulus is going to consist of glomerular capillaries as well as several supporting cells. And this is going to be part of the renal corpuscle. And the glomerular capillaries are then going to be drained by a second arteriole, and this one's called the efferent arteriole. And the efferent arteriole feeds a second capillary bed, which you saw, called the peritubular capillaries. All of this together forms a plexus or a network around the renal tubule of each nephron. And the order of this system, this system of capillaries is as follows. You have the afferent arteriole feeding the glomerulus, which drains into the efferent arteriole, which drains into the peritubular capillaries, and then the venules carry that out. So there is a venous path. And here's that image one more time. So we're just going to look at the arterioles this time. So you see coming off of this this interlobular artery, we have the afferent arterioles here feeding into the glomerulus, and then the glomerulus feeds the efferent arterioles. The efferent arterioles then feed these peritubular capillaries, which go into the venous path now. The venous path is essentially going to be the parallel of that arterial path. Groups of peritubular venules are going to unite into a progressively larger venous vessels. So it goes interlobular vein, arcuate vein, interlobar vein, renal vein. You're going to notice here that I did not talk about segmental veins because they do not exist. Interlobar veins merge in the renal sinus and form that large renal vein. And the renal vein is then going to exit the hilum, and empty directly into the inferior vena cava. And the venous path, we just sort of track as you see this interlobular vein merging into the arcuate vein. And then we zoom out, the arcuate veins join into interlobar veins here, and the interlobar veins all join back up into the renal vein. And then that joins up with the inferior vena cava, draining the kidney of blood, of now filtered blood, to be precise. So as we just talked about, most of these functions that the kidney does are going to occur in their tiny nephrons. And the nephrons filter the blood, and they modify the filtered fluid as it passes through their renal tubules. The tubules, we're going to talk about the nephron. It has two major divisions. You have the renal corpuscle and the renal tubule. The renal corpuscle is responsible for all of that filtering of the blood. Each globe-shaped corpuscle consists of two parts. You have the glomerulus and the outer sheath of epithelial tissue called the glomerular capsule, or a Bowman's capsule. Same thing. The glomerulus is a looping group of fenestrated capillaries. What does that mean? It means they have very large pores present within their plasma membranes, as well as in between their endothelial cells. So these fenestrations, this porous structure, makes these capillaries extremely leaky or permeable, and it forms a main part of the filtering structure of the renal corpuscle. So this is a good look at that renal corpuscle, and you can see these arterioles here. They are just extremely porous. In this artistic drawing, you can see it's sort of it looks like slits, and then on the scanning electron micrograph here, you can actually see all of those filtration slits. That's what they're actually called. On the outside of the glomerulus, you're going to find that it is a double-layered glomerular capsule, and this consists of an outer layer and an inner layer. There's the parietal outer layer and the inner visceral layer, just like the peritoneum, outer parietal, inner visceral. The parietal layer is really going to be a globe-like extension of the renal tubule, and it consists of simple squamous epithelial tissue. The visceral layer, on the other hand, is going to consist of specialized epithelial cells called podocytes that wrap around the glomerular capillaries. And each of these are going to extend off of them, and they're going to have what are called foot processes or pedicels. And these pedicels weave together to form those filtration slits, which make up the other part of the renal corpuscle's filtering structure. Between the parietal and the visceral layers, you have a hollow region, a lumen, and this lumen is also going to be called the capsular space. This capsular space is continuous with the beginning of the renal tubule. Once the podocytes and the fenestrated glomerular capillaries are together, they form a part of a complex membrane that filters the blood as it flows through the glomerulus. This structure allows a great volume of blood to be filtered, and that fluid, as it passes through the filter, leaving the glomerular capillaries, it is now known as filtrate, as it first enters that capsular space, and then it's going to flow out the capsular space into the lumen of the renal tubule. And you can see here some of this filtrate exiting or being filtered out through the renal tubule here. You can see it just being filtered out, and it will enter it will exit this glomerulus into the actual lumen of the renal tubule there. So these are called the quote unquote pipes of the nephron, and this renal tubule is basically just a winding tube that modifies that filtrate. So we filtered this fluid out of the blood, now we need to modify it. And the renal tubule has three regions. There's the proximal tubule, the nephron loop, and the distal tubule, each of which is going to differ in structure and function. This first region here you see is the proximal tubule. What does it mean? It means it's proximal. It's the first region. It's the closest region to the point of origin, right? And the origin is the glomerulus. So the first and longest segment is the proximal tubule. And this, of course, has simple cuboidal epithelial cells. You see here, simple cuboidal cells. And those all have very prominent microvilli. This part of the tube has been coiled or convoluted, but it also has several straight sections. And that's why we're basically going to talk about the proximal tubule rather than the proximal convoluted tubule, because it has convoluted and straight areas. Now, all of those microvilli that these cuboidal cells have, they're going to form a brush border. And we talked about brush borders with the enterocytes. This is pretty much doing the same thing. These are going to greatly increase the tubule's surface area, allowing it to better filter the blood, the filtrate. Now, you have the nephron loop. This is where that filtrate flows out of the proximal the proximal tubule into the nephron loop, also called the loop of Henle. And this is the only part of the renal tubule that will dip into the renal medulla. And the nephron loop tends to have two limbs. It has a descending limb that travels toward the medulla, and then it turns 180 degrees and it becomes the ascending medulla. The ascending limb, and it climbs out of the medulla back into the cortex. Most of this limb, the descending limb, is going to be made up of simple squamous epithelium, and so that simple squamous epithelium leads it to be called the thin descending limb. And in some nephrons, this thin segment also forms the bend region and part of the ascending limb. So there is also a thin ascending limb. But most of the ascending limb is composed of thicker cuboidal epithelium and can be then referred to or distinguished as the thick ascending limb. And we can see that here, this with that transfer from simple squamous to this thick ascending limb that is now made up of cuboidal epithelium. Notice it does not have any microvilli on it. And again, we're looking at this generalized nephron and collecting system. So you see you have the renal corpuscle with the glomerulus inside, feeding that proximal tubule, and eventually you get to the nephron loop here. So you have the descending loop here, and then you turn around and have the ascending loop, but then the ascending thin loop, and then you have the ascending thick loop, okay? All of that feeding into the distal tubule. And the distal tubule has this final segment has simple cuboidal epithelium. However, this one has much sparser microvilli, and so this doesn't really make up a brush border, but those microvilli do still work to increase the surface area. And you can see those here. You can see the microvilli present here. They're not as uniform, and they don't really resemble the bristles of a brush as much anymore. And so here you can see all those put together. The proximal tubule with its brush border. You get down to the thin descending limb with this simple squamous epithelium. You get the thin ascending limb, and then you get to the thick ascending limb here, which has no microvilli but is still cuboidal epithelium tissue. And that will eventually feed into the distal tubule, which has limited microvilli, not a brush border. So it all seems like it might run together, but you got to remember this combination of cell type and microvilli presence to really make sure you get these points hammered home. Now, where the ascending limb of the nephron loop meets the distal tubule, there's going to be a tightly packed group of cells called the macula densa, the dense spot. And this macula densa comes into contact with what are basically smooth muscle cells in both the afferent and efferent arterioles, known as juxtaglomerular cells. Together, the macula densa and the juxtaglomerular cells form a structure called the juxtaglomerular apparatus, and this JG apparatus regulates blood pressure as well as glomerular filtration rate. We're going to look at this again when we get to section 4 here. But here's what that apparatus looks like. You can see this macula densa contacting those efferent arterioles, and the afferent arterioles, and in addition to that, you have those you see here, these juxtaglomerular cells, which are basically muscle cells, and all of those work together, controlling that glomerular filtration rate. And the nephron is really going to end basically where the filtrate in the system, in the distal tubule, is going to empty into what is called the collecting system. So the nephron has done all this filtration, and now it dumps the fluid, or the filtrate, into the collecting system. And these are all functionally distinct tubules, further modifying the filtrate as it passes through them. Most of these are going to be basic cuboidal or columnar cells with very few microvilli. And in this image here, you see it's just cuboidal epithelium. But it could also be a columnar epithelium, which basically, if you remember, just means they're longer. And the collecting system consists of the cortical collecting duct and the medullary collecting system. And the distal tubule is going to empty that filtrate into the first part, called the cortical collecting duct. And this is found within the renal cortex. The cortical collecting duct, again, is going to be made up of those cuboidal cells. Do note that each cortical collecting duct drains several distal tubules. You'll see that they're the sort of universal dumping grounds for these distal tubules. And as the cortical collecting duct passes down into the renal medulla, it then changes names into the medullary collecting duct. And deep within the renal medulla, several medullary collecting ducts empty that filtrate into those larger papillary ducts. And the papillary ducts are going to contain very low columnar epithelial cells, and they include the medullary collecting system includes medullary collecting ducts as well as the papillary ducts. Once the filtrate has reached the end of the papillary duct, it is now called urine, and the urine exits the papilla of the renal pyramid into a minor calyx, and any formation of crystals within the tubules can block this flow of this filtrate, and it can lead to intense pain. Those are called kidney stones, right? The technical term is nephrolithiasis. And here you can see that sort of structure all put together. You see the cortical collecting duct here in the center, and it consists of this whole collecting system. And then once it enters the renal medulla, it becomes the medullary collecting duct. And the medullary collecting duct just drains down and enters the papilla, the renal pyramid there, and it becomes the papillary collecting duct. So this is sort of the same structure, just changing names based on where it is. Speaking of kidney stones, the condition is referred to as nephrolithiasis, but the actual stone is actually called a renal calculus. A renal calculus is basically going to be a crystalline structure composed mostly of calcium oxalate salts. And these form when the concentrations of any of these calcium oxalate ions really are higher than normal. This can be as well as solutes like sodium, hydrogen, and uric acid ions. This condition is basically called supersaturation, and supersaturated ions are more likely to come out of solution and crystallize. There are a number of risk factors for this supersaturation, like dehydration. If there's not enough water in the diet, those supersaturated solutes are going to come out. High fat diets, animal proteins, salts, and obesity. Typically, these crystals are going to form in the nephron loop, in the distal tubule, and or the collecting system. And the truth is most crystals simply pass unnoticed in the urine. However, sometimes these crystals are going to adhere to the epithelium of the tubules, especially in the collecting system, and they form what are called seed crystals. And these seed crystals can lead to the formation of stones. And these stones then remain in the collecting system, or they break off and are lodged then in the calyces or the renal pelvis or the ureter. Any stones lodged within the urinary system cause the most common symptom, severe pain, known as renal colic. And renal colic radiates from the lumbar region to the pubic region, and any other symptoms, include blood in the urine, sweating, nausea, vomiting. Typical diagnosis involves some CT scans or some intravenous pyelograms or an IVP. An IVP is basically a radiograph of the urinary system that uses a contrast like iodine. So it's basically an X-ray of the urinary system, and they use iodine because iodine prevents that radiation from passing through. And we're going to look here at an IVP scan. You see the urine in the urinary bladder is very, very visible, so it's blocking a lot of that light. And this patient here has a renal calculus blocking their left ureter. So you can see that they're not getting, that there's an irregularity shown up there. I believe that maybe just highlighted in yellow, but it is still an irregularity. And so that would be the renal calculus. Now, basically everything I just said was sort of a generalized version of a nephron, right? And I remember I said there are two types of nephrons based on the structure and functions of their nephron loops. These two types are labeled cortical nephrons and juxtamedullary nephrons. Now, about 80% of the kidney's nephrons are going to be cortical nephrons, named so because they are primarily in the renal cortex. The renal corpuscles of these cortical nephrons are located in the outer renal cortex and have very short nephron loops that either just dip into the superficial part of the medulla or they never leave the cortex at all. You have peritubular capillaries supplying the blood to the nephron loops of these cortical nephrons here. As opposed to that, you have juxtamedullary nephrons, and these are less numerous. And you're going to notice that the renal corpuscle of a juxtamedullary nephron sits very close to that cortex medulla boundary. In addition, it has a very long nephron loop burrowing very deeply into the renal medulla. Inside of this, the loop is going to be surrounded by a ladder-like network of capillaries called the vasa recta, or straight vessels. And these arise from what is essentially the efferent arteriole. And much like the peritubular capillaries, the vasa recta empty into interlobular veins. And notice, when we look at this photo, the portion of the nephron that lies in the cortex is going to be surrounded by several branches of regular peritubular capillaries. This unique structure allows those juxtamedullary nephrons to function as part of a system that controls the volume and the concentration of urine. We're going to talk about that when we get to chapter 6, or part 6 of this chapter. And here you can see some of those differences in the structure. You see the afferent arteriole feeding into the glomerulus, and then taking out the efferent arteriole, taking that blood out, and then feeding all of these tubules, as well as the nephron loop, with the capillaries here. This peritubular capillary just running all across these tubules. And this is all in the cortical nephron here. A way to remember this is if you see these arcuate arteries and veins, basically these run along the curvature of a renal pyramid, right? So everything down here is renal medulla, everything up here is renal cortex. And so cortex, cortical, that's a good way to remember it. And then you have juxtamedullary, and these juxtamedullary nephrons are going to dip way down into the medulla, and you can see that the proximal tubule and these glomerulus, all these proximal tubule and distal tubule, they're all fed by the peritubular capillary, but the nephron loop itself is fed by the vasa recta. So that's a little bit of a difference there. Everything up here fed by these peritubular capillaries. You have the distinction here of the vasa recta in the juxtamedullary. So up until this point, we've basically talked about anatomy. Now let's talk about the physiology here of the urinary system. These nephrons are going to, and the collecting system are going to carry out three basic processes that allow the kidneys to perform their functions. They filter, they reabsorb, and they secrete. So the first thing that happens is the glomerular filtration. And this is how the the nephrons filter their blood. And this takes place as the blood passes through those glomerular capillaries, and some of the plasma is filtered into the surrounding capsular space. The endothelial cells of the glomerulus form, remember, they're that very, very porous size, and so they're enabling them to hold back cells and proteins, but allow lots of water, electrolytes, lots of water and ions and acids and bases to basically come out, along with organic compounds, metabolic wastes, stuff like that. All that exits the blood, and it's going to enter that glomerular capsule now. And then the fluid and the solutes now are going to be the filtrate. Now they're no longer plasma or wastes or whatever. They're now all known as filtrate, also known as tubular fluid. This tubular fluid is then going to be modified as it flows through the tubules. And much of this is really going to be involving reclaiming substances like water or glucose, amino acids, electrolytes, anything that we need that's in this filtrate. And it's going to return those solutes to the blood. And this process is called tubular resorption. Tubular reabsorption, I'm sorry, because it's reabsorption that occurs in the tubules. It's very, very simple. I totally forgot to hit forward on that. I'm sorry. So the majority of this water and these solutes that are filtered through the glomerulus are going to be reabsorbed. And most of this reabsorption is going to be in the proximal tubule and the nephron loop. But some very specifically controlled reabsorption also occurs in the distal tubule and the collecting system. This really just allows the kidney to vary the amount of different substances that are reabsorbed to maintain homeostasis. So this specialization allows for that, for specific things to be filtered out in specific places. Now, you also have tubular secretion, and this filtrate is going to be modified by tubular secretion, and that's essentially the reverse of tubular reabsorption. And notice that in a figure here we show in a minute, you're going to see substances moved from the peritubular capillary blood into the filtrate in the tubules, and it eventually gets excreted. Secretion happens in pretty much all the parts of the nephron and the collecting system, although, of course, different substances may be secreted more in one area than another. So it's another sort of specialization. And this also helps maintain electrolyte and acid-base homeostasis, as well as removing any extra toxins from the blood that may not have entered during filtration. So here you can see this image here. You see filtration, resorption, and secretion. And if I say resorption instead of reabsorption, it's the same thing. So you see filtration occurring in the renal corpuscle through the glomerulus. That's the only place that happens. Then you have resorption taking place in the proximal tubule, in both limbs of the nephron loop. You also have it happening some in the distal tubule and the collecting duct. The same can be said for secretion in both tubules and the collecting duct. So substances get secreted back into the blood, or I'm sorry, back into the solute from the blood in the proximal tubule, the distal tubule, and the collecting duct, and they get reabsorbed everywhere, basically. So this is a pretty handy figure if you guys need to study something. This would be a good one. Where does filtration occur? Where does reabsorption occur? Where does secretion occur? Now, filtration is really easy to think about when you cook spaghetti. You take the big pot full of water and pasta noodles and you dump it into a colander, and you filter the fluid and the solutes from each other. The solutes in this case would be the spaghetti, and the water would be the extra fluid that we get out of it. So we're passing small substances through a filter and leaving large substances behind. And this is essentially what's happening in the kidneys. This membrane, instead of being a colander, is now going to be the filtration membrane, and it forms a filter that allows that filtrate, so those fluids and ions, to pass through, but it leaves large substances behind in the blood. In this section, we're going to look at the structure of that membrane, the mechanical process of filtration, and several factors that determine the rate at which filtrate is formed. So the inner layer, remember the inner layer of the glomerular capsule has these 3 layers together called the filtration membrane. It's made up of glomerular capillary endothelial cells, a basal lamina, and the podocytes, that visceral layer of the glomerular capsule. So let's look at these from deep to superficial. Basically, this is the same direction that filtrate will flow. So first things first are the fenestrated glomerular capillary endothelial cells. And if you guys remember, just like all capillaries, these glomeruli are going to be composed of epithelial cells, but they're fenestrated. They have large pores that make them leakier than most capillaries. And the gaps are pretty large, about between 70 and 100 nanometers. So that's pretty big. They're still small enough to prevent red blood cells and platelets from going out, but lots of water and ions are really readily able to go through. Next, you have the basal lamina, and this is a thin layer of an extracellular matrix gel that separates the glomerular endothelial cells from the podocytes. There's a lot of collagen fibers here that basically form a mesh that acts like that colander, and it prevents anything greater than 8 nanometers from getting into the capsular space, basically blocking most plasma proteins. They also have a negative charge that repels anything negative. Again, these are for plasma proteins. And this occurs even in things that are larger, or smaller rather, than 8 nanometers. So that charged, that really small charged porous membrane called the basal lamina is another filtration membrane. You also have the podocytes composing that visceral layer of the glomerular capsule. So this inner layer making up the third and finest filter. Now, these are going to wrap around the glomerular capillaries and they're going to interlace. So just kind of weave your fingers together, and that's sort of what it looks like. These narrow slits only allow things that are less than 6 to 7 nanometers. They only allow things like that are things that are that small or smaller to enter this capsular space, less than 7 nanometers. And albumin is essentially the biggest, by composition, albumin is the most prevalent, is a better way to say it, plasma protein. And albumin is about 7.1 nanometers. So these podocytes really work together to keep out most of this albumin, and it normally prevents it from entering any of that filtrate. So from deep to superficial, let's start on the right-hand side of the image here. You can see these red blood cells right here are too big to go through any of these capillary walls, any of these fenestrations here in the capillary walls. You see, these are those fenestrations I'm talking about. Now, here's another one. You also see, as you look over here on the left-hand side, a sort of zoomed out version. You can also see these podocytes, these foot processes here, as they come down and branch out. They interlace. You can see some of that here. All of that forming yet another filtration membrane. But if you look back over on the right, you're going to see an even smaller membrane here. This filter that runs the entire length all the way down, and it's the finest layer. This is the basal lamina. And so you can see as you get through here, you got the podocytes, you've got the fenestrated capillaries here, you've got the basal lamina here, and it's all just working together to be a finer and finer mesh, preventing things that we want to stay in our blood from getting out, while still allowing, our water and our small solutes like hydrogen ions from getting out. No, it still allows them out. I'm sorry, it allows them to get out, preventing them from stinking up our blood. Now test yourself really quick. Which of these is not a part of the filtration membrane? Which one is not part of the filtration membrane? The macula densa is not part of the filtration membrane. That's part of the tubules, and it helps control that glomerular filtration flow, but is not part of it itself. The composition of the filtrate is based primarily on what? Based primarily on size of the particles. So the size of those particles is really what makes the decision whether it's going to be part of the filtrate or if it's going to stay in the blood. Now, which of these does not normally appear in the filtrate? You got water, urea, albumin, and glucose. Well, one of those is what most of the filtrate is, right? So it's not that one. It's albumin. Albumin is typically too big to go through that basal lamina, remember? So this is that least permeable membrane of the filtration membrane. The least permeable. So it's got the smallest pores. It's the podocytes. Those foot processes make up a 6 to 7 nanometer mesh, really making it really, really hard for things to get through. Even the albumin proteins are stuck in the blood. All right, that's going to be it for this slideshow. I'll see you guys in the next one.